welcome to Biology 40C. Today we're going to talk about acid-base balance. That's chapters 25 and 26 of your textbook. Let's start by watching this short video about the history of Gatorade.
They said it was an experiment. I don't believe there's any modern place in the world, Florida Field on a Saturday afternoon. It was built over a swampy area just to the side of campus there. And gosh, I remember being dead tired after warm-up. There's always that threat of dying. I mean, you have no idea how brutal the heat can be.
We put 20 football players in the infirmary.
And that was really what set off this whole problem-solving quest.
All right, we've identified the problem. Now, what is the solution?
We all had a bond because we went through this experiment together.
We thought we had an edge.
We felt like we had a magic elixir.
You never know when that small idea is going to take wings and literally change the course of history.
So why did 20 football players end up in the infirmary? What's the physiology that's going on here? Well, along with their sweat losses, their water losses, there were losses of electrolytes. For example, losses of sodium, losses of potassium, and some losses of calcium and magnesium. Sodium is particularly important to maintain fluid balance, to maintain nervous system and muscle function. And potassium is essential for regulating electrical gradients that are essential for muscle contraction. Calcium is also involved in muscle contraction, and magnesium can help with muscle relaxation. So what's going on in the kidneys of players that are drinking just water, that are trying to prevent dehydration by drinking water? What happens is that excess water dilutes the sodium in the blood, reducing the ability of the kidneys to retain water, because this osmolarity gradient in the loop of Henle is what's essential for allowing the kidneys to retain water. Water moves across into this high salt environment here and out of the urine. So what's happening to players that are chugging water? After chugging water, the excess water dilutes the sodium in the blood. As that electrolyte concentration drops, the sodium-potassium gradients are reduced, and as that happens, the nerves which rely on a sodium-potassium gradient and sodium-potassium pumped to maintain the electrical gradients across the nerves can no longer repolarize. And when they can no longer repolarize, the muscles cannot relax, and that's called cramping. So the key electrolytes involved in muscle cramping are sodium, potassium, calcium, and magnesium. Sodium is essential for muscle contraction and creating the electrical gradient along with potassium. Calcium triggers contraction by interacting with muscle proteins, and magnesium promotes relaxation by regulating the attachment of ATP to the muscle fibers. So electrical imbalances are generally caused by low sodium or disturbances in sodium and potassium concentrations. This disturbs the ability of the muscle to contract by altering the nervous system signaling, and this leads to an involuntary cramp. Low magnesium impairs calcium control, and that can also cause excessive contraction. Low magnesium really results in an inability of the muscle to relax. And overall, this is caused by loss of electrolytes through sweat. There are fluid spaces, extracellular fluid spaces, and intracellular fluid spaces. And these two need to remain in balance. In the case of hyponatremia, hypo meaning low, natremia meaning sodium. If you look at the periodic table of elements, Na is the letter for sodium. It's a German word meaning sodium, natrium. So hyponatremia means low sodium. So if in the extracellular fluid, you get low sodium. This could occur because of a high sweat rate and a lot of sodium losses through sweating. How is that going to affect the flow of water from the intracellular from the extracellular to the intracellular space? And why might this lead to a headache, for example? What you would imagine, what you would see happening is that the fluid, the water in the extracellular fluid, will flow from low concentration to high concentration, where there are more electrolytes, into those intracellular fluid spaces. This can cause cells to swell. Obviously, if cells are swelling, that's a bad thing. And if this is happening to cells in your brain, neuronal cells, this can cause headache. So this would be osmosis causing brain swelling. The skull is very hard, and so the brain has no room to expand. So as the brain expands due to swelling with this water, it will start to press out against the skull, and that will cause a severe headache and can ultimately result in death. Different electrolytes are found inside and outside of cells. What that does is that maintains a electrochemical gradient across the membranes of cells. So if you look at plasma, there's a lot of sodium in blood plasma. In extracellular fluid, there is a lot of sodium and chloride ions balancing that. There's obviously our bicarbonate buffer there as well. But within cells, the situation is quite different. Within cells, there's very low levels of sodium, but very high levels of potassium. This occurs because there is a sodium-potassium pump where sodium is pumped outside of cells and potassium is pumped inside of cells. When this happens, the pump is asymmetric and it results in more potassium inside of cells and less sodium outside of cells, creating a small electrochemical difference, much like a battery. So this membrane potential can be used for all kinds of different functions within the cell, but in nerve cells, this will be used for firing the nervous system. And muscle cells, this can be used to fire the muscle cells and contract muscle cells. Most of the fluid in sweat comes from the extracellular fluid, high in sodium and low in potassium. So the main electrolyte that we're concerned about in hot conditions, in conditions of sweating, is sodium. Sodium losses are the main losses that you'll see. So why does Gatorade work? Gatorade is different from water because Gatorade contains two really crucial components. It contains glucose and it contains sodium. Both glucose and sodium are co-transported into the body on a co-transporter molecule SGLT2. SGLT2 allows sodium and glucose uptake. Glucose is then transported into the blood and can be used as a source of energy. And the sodium is transported in the opposite direction of potassium and enters the bloodstream. So sodium and potassium are now regulated here together. The sodium enters the bloodstream and helps to balance the lost electrolytes, the electrolytes that are being lost through sweat. One of the most popular recent trends is this idea of drinking alkaline water. We're going to talk about this in class, and I want you to have a little bit of a think about whether alkaline water is something that could be good for your body or not, and why. Alkaline water hits us about here, about a pH of 9, where the body's pH is about 7. I will remind you that the stomach is somewhere around pH 2 to 3. I will watch this video in class. You should go to this website and review it if you would like to see it again. One of the key mechanisms that the body requires uh, for regulation of total body pH is this carbonic acid bicarbonate buffer system. This is one of the key mechanisms by which the body maintains its acidity and basic nature to remain at a very neutral, very close to neutral pH at all times. The way the body does this is through a number of different chemicals, and central to um, the system is the carbonic acid bicarbonate system. So the bicarbonate ion in the blood um, can combine with hydrogen ions to form carbonic acid. And then carbonic acid in the lungs can result, it can dissociate into carbon dioxide and water. And the lungs can then blow off carbon dioxide. Uh, in general, bicarbonate is stored in reserves as sodium bicarbonate and will only combine with hydrogen ions when blood acidity increases. That might be through having a meal that's got high in acid content, but more likely it's from alterations in pH in the blood through activity. And one of the key activities that can drive up blood acidity is through the production of lactic acid. And lactic acid is produced when exercising with when exercising at high intensity before oxidative phosphorylation has caught up, and when you're breaking down glucose. So let's take a look at what happens with metabolic acidosis and respiratory exchange, blowing off, if you will, excess carbon dioxide to help your body to maintain its pH balance.
What actually happens when acid is introduced into the body? Well, buffers take care of it. What are buffers? Buffers act like sponges, taking up hydrogen ions when there's too much of them and releasing them when there's too little. The primary buffers in the body are bicarbonate, or HCO3 minus, hemoglobin, phosphate, and bone. 60% of buffering happens intracellularly, and 40% happens extracellularly. The primary buffer in the extracellular compartment is bicarbonate. Let's see what happens when acid is added into the body. Let's talk about metabolic acidosis. So, in metabolic acidosis, there's an excess of hydrogen ions. And what happens to these hydrogen ions? They combine with bicarbonate in order to form carbonic acid. So as hydrogen ions increase, bicarbonate decreases. And what happens to carbonic acid? Well, that dissociates into water and carbon dioxide. And what happens to carbon dioxide? Well, carbon dioxide, as you might already know, is then eliminated and exhaled through the lungs. We can adjust carbon dioxide elimination through hyper and hypoventilation. For completeness' sake, it's important to understand that this equilibrium also works into the other direction. So when the patient is hypoventilating, carbon dioxide is accumulating and combines with water to form carbonic acid, which then dissociates into hydrogen ions and bicarb. So hydrogen ions will go up in hypoventilation and the pH will consequently drop. You'll learn more about this equilibrium and the association between the respiratory and the metabolic system in the upcoming modules.
So there you have it. This system gives us near instantaneous pH balance. When carbonic acid dissociates to form carbon dioxide and water, we can blow off this extra carbon dioxide, and that will shift this equilibrium. There's an enzyme involved called the carbonic anhydrase. We can shift this equilibrium so we can instantaneously remove hydrogen ions, turning by using bicarbonate, turning that into carbonic acid. Carbonic acid, with the help of carbonic anhydrase in the lungs, can help us remove carbon dioxide. So what happens when you exercise really hard and you make a lot of lactic acid? Well, when you make lactic acid, the acid component of lactic acid here is the hydrogen ion. And what you will notice that when you work out really hard in a sprint and you produce a great deal of lactic acid, one of the first things that the body does is it starts hyperventilating. In the blood, we have our carbonic acid buffering system, which is nearly instantaneous by taking hydrogen ions, combining them with bicarbonate ions into carbonic acid, and then that carbonic acid is dissociated into carbon dioxide and water. This system can happen very, very quickly. So let's think for a moment. If we drink alkaline water, assuming alkaline water could bypass the stomach, where it would be instantly, the pH would be instantly neutralized. But let's say, for example, that the person is on a drug that results in their stomach pH not being normal, an omeprazole or some other kind of acid reducing drug. And so now the alkaline water might momentarily decrease hydrogen ions in the blood. That would shift this equilibrium towards more carbonic acid and less carbon dioxide and water to blow off. So that could cause a momentary decrease in um, carbon dioxide necessary to blow off. So this would basically reduce your breathing rate by driving the reaction to the right. It's less carbon dioxide, and you would increase your hydrogen ions to balance any decrease in pH from the water. Now, this process would be nearly instantaneous, and your blood pH would return, blood acidity would return to the levels where they started almost immediately. If, on the other hand, you eat something very acidic, and this again could bypass your stomach acid, which generally it can't, the acidity will drive the reaction to the left, increased hydrogen ions, driving the reactions to the left, increased hydrogen ions soaked up by the bicarbonate buffer, forming carbonic acid. Carbonic acid will dissociate in the lungs into carbon dioxide and water. After a little bit of quicker breathing, your blood pH will return to normal. This is a very quick process. Carbon dioxide dissolved in the plasma can be excreted in the alveoli. If this is happening in a red blood cell, again, the carbonic anhydrase in red blood cells will shift and produce that carbon dioxide, which will rapidly dissociate. And in the red blood cells at the same time as carbon dioxide is being removed and being carried on hemoglobin here, carbon dioxide being carried on hemoglobin to the lungs where it's going to be removed. Oxygen will be displacing that carbon dioxide, and then that oxygen can be delivered to the muscle cells where it's going to be effective for glycolysis and producing ATP energy. So I'm going to ask you to work on a card where a friend tells you that they heard that most people's bodies are too acidic these days and they need to drink alkaline water. I hope from these slides that you can see that that is very unlikely to be the case. If a person's body were too acidic, they'd be dead. So I would like you to refute your friend's claims and include in your response information about the buffering system, how it works, and particularly in the case where the body is overly acidic, and information about the activity of the lungs, and also give to me what the kidneys at the same time will be doing to buffer an increase in pH. How are the kidneys going to also buffer and remove hydrogen ions, those acidic hydrogen ions? So let's take a look here at the role of the kidney in acid-base balance. So we've seen the role of the lungs. Now let's take a quick look at the role of the kidney.
The kidneys have two main ways to maintain acid-base balance. Their cells reabsorb bicarbonate, or HCO3-, from the urine back to the blood, and they secrete hydrogen ions into the urine. By adjusting the amounts reabsorbed and secreted, they can balance the bloodstream's pH. Now, our kidneys filter blood continuously by distributing the blood that comes into the kidney to millions of tiny functional units called nephrons. Each nephron is made up of a glomerulus, or a tiny clump of capillaries, where blood filtration starts. When blood passes through a glomerulus, about 1/5 of the plasma leaves the glomerular capillaries and goes into the renal tubule. The renal tubules function to reabsorb the good stuff, like water and electrolytes, and leave behind the bad stuff, like waste products and acid. The renal tubule is a structure with several segments: the proximal convoluted tubule, the U-shaped loop of Henle with a thin descending limb and a thick ascending limb, and the distal convoluted tubule, which goes back up again before emptying into the collecting duct, which collects the final urine. Each of these tubules is lined by brush border cells, which have two surfaces. One is the apical surface that faces the tubular lumen and is lined with microvilli, which are tiny little projections that increase the cell's surface area to help with solute reabsorption. The other is the basolateral surface, which faces the peritubular capillaries, which run alongside the nephron. So with bicarbonate reabsorption, as the filtrate leaves the glomerulus, it first goes through the proximal convoluted tubule. At first, this filtrate has the same concentration of electrolytes as the plasma it came from. But when a molecule of bicarbonate approaches the apical surface of the brush border cell, it binds to hydrogen that was secreted by the brush border cell in exchange for a sodium ion. And this forms carbonic acid. At this point, an enzyme called carbonic anhydrase type 4 that lurks in the tubule in the microvilli, kind of like a shark, swims along and splits the carbonic acid into water and carbon dioxide. Unlike charged bicarbonate anions, which are stuck in the tubule, the water and carbon dioxide happily diffuse across the membrane into cells, where carbonic anhydrase type 2 facilitates the reverse reaction, combining them to form carbonic acid, which dissolves into bicarbonate and hydrogen. A sodium bicarbonate co-transporter on the basolateral surface snatches up the bicarbonate and a nearby sodium, and then shuttles both into the blood. Alternatively, a bicarbonate chloride exchanger exchanges bicarbonate with chloride, leaving the bloodstream to enter the cells. All this chemical trickery effectively moves 99.9% of the filtered bicarbonate that's in the tubule back into the bloodstream. Now, hydrogen ions, with their positive charge, don't want to naturally pass through the cell membranes and into the urine. They need to be pushed out. And there are two mechanisms that do this. One mechanism is a sodium-hydrogen countertransport. With this, a carrier protein in the apical wall binds a hydrogen ion from the cell and a sodium ion in the tubular fluid. As that sodium ion is pushed in, the hydrogen ion gets pushed out, kind of like a revolving door. Remember, this is in the proximal tubule, but in the distal tubule and collecting ducts, there's another mechanism that involves alpha intercalated cells. These cells have a different pump that uses the energy of ATP to push hydrogen ions into the tubule. The urine, though, can only hold so many free hydrogen ions because the pH starts to drop really low, and the tubules can't maintain a urine pH below about 4.5. So to get around this limit and hold more hydrogen ions, the urine has chemical buffers, which bind to the hydrogen ions and prevent the pH from dropping too low. The most important is the ammonia buffer system, in which the kidneys use a process called ammoniogenesis. Ammoniogenesis starts when the proximal convoluted tubule cells break down amino acids like glutamine into ammonia, or NH3. The ammonia is lipid soluble, so it diffuses freely into the tubule, where it combines with a hydrogen ion to form an ammonium ion, or NH4+. Ammonium combines with chloride in the urine. And because ammonium chloride is only weakly acidic, the urine pH doesn't drop much, even though it has a lot of hydrogen ions. Most of this ammonium is lost in the urine, which helps the kidneys get rid of a large amount of hydrogen. A second buffer system uses phosphate. Monohydrogen phosphate, or HPO4 2-, enters the tubule from the plasma. Monohydrogen phosphate's poorly reabsorbed from the tubules, so it ends up concentrating there. It acts as a buffer by combining with secreted hydrogen ions to form dihydrogen phosphate, or H2PO4-, which is then peed out in the urine. All right, as a quick recap, the kidneys help maintain pH balance of the blood. In the nephron, the proximal convoluted tubule cells are able to reabsorb the bicarbonate ions, and cells in the proximal, as well as the distal convoluted tubule and collecting ducts, secrete hydrogen ions that are carried out into the urine using the ammonia and phosphate buffer systems.